on some aspects related to immunology, like cell-mediated effector function, myeloid, lymphoid, and secondary lymphoid organs. So, cell-mediated effector functions. Clonal selection theory. The theory states that in a pre-existing group of lymphocytes, specifically B cells, a specific antigen only activates. That means selection. Its counter-specific cell, so that particular cell is induced to multiply, producing its clones for antibody production. This activation occurs in secondary lymphoid organs such as the spleen and the lymph nodes. Then clonal selection of lymphocytes. A hematopoietic stem cell undergoes differentiation and genetic rearrangement to produce immature lymphocytes with many different antigen receptors. Those that bind to antigens from the body's own tissues are distinct, while the rest mature into inactive lymphocytes. Most of these will never encounter a matching foreign antigen, but those that do are activated and produce many clones of themselves. And two types of cells are produced by clonal selection: effector cells, short-lived activated cells that defend the body in an immune response; then memory cells, cell-mediated effector functions. The effector cells of the cell-mediated immune system include cells of the innate immune system, natural killer cells, cells of the adaptive immune system, helper CD4 T cells, TH cells, and CD8 cytotoxic T lymphocytes, CTLs or TC cells. Then NKT cells are also participants and have features of both the innate and adaptive immune systems. Effector cytotoxic cells arise from both the adaptive and innate immune systems and therefore include both antigen-specific and non-specific cells. Antigen non-specific innate immune cells that contribute to the clearance of infected cells include NK cells and non-lymphoid cell types such as macrophages, neutrophils, and eosinophils. Then antigen-specific cytotoxic cells include CD8 T lymphocytes, CTLs or TC cells, as well as the CD4 NKT cells, populations of cytotoxic CD4 TH cells may contribute to delayed type 2 hypersensitivity. Then how cytotoxic lymphocytes act. So cell-mediated immunity consists of both helper T cells, CD4-TH, and several types of cytotoxic cells. Myeloid progenitor cells, granulocytes often called polymorphonuclear lymphocyte. They are classified into various forms on the basis of differences in cellular morphology. Neutrophils have multi-lobed nucleus and granulated cytoplasm. Movement of circulating neutrophils into tissues called extravasation. Then eosinophils, bilobed nucleus and stained with eosin. They defend against protozoan and helminth parasites by releasing cationic peptides and reactive oxygen intermediates into the extracellular fluid. Then basophils, stained with methylene blue. They are non-phagocytic, release substances that cause an allergic response. These molecules include histamine, prostaglandin, serotonin, leukotrienes. Basophils possess high affinity receptors for IgE. Then monocytes, mononuclear phagocytic leukocytes that circulate briefly in the bloodstream before migrating into tissue where it becomes macrophages or into dendritic cells. They migrate into the tissue and differentiate into specific tissue macrophages or into dendritic cells. Macrophages. Inflammatory macrophages are expert phagocytes and typically participate in the innate immune response. They have dual role: contribute directly to the clearance of pathogens from a tissue, act as antigen presenting cells. Tissue resident macrophages include Kupffer cells in the liver, microglia in the brain, and alveolar macrophages in the lungs. Their role as antigen presenting cells. Then lymphoid organs. Innate immune cells mount the first line of defense against pathogen. Antigen presenting cells communicate the infection to lymphoid cells, which coordinate the adaptive response, generate the memory cells, and prevent future infections. The coordination required for a full immune response is made possible by the specialized anatomy and microanatomy of the immune system. called lymphoid organs, which remain dispersed throughout the body. Primary lymphoid organs include bone marrow and the thymus, are sites where immune cells develop from immature precursors. And secondary lymphoid organs include spleen, lymph nodes, and specialized sites in the gut and other mucosal tissue. Sites where the mature antigen-specific lymphocytes first encounter antigen and begin their differentiation into effector and memory cells. In primary lymphoid organs, where immune cells develop. HSCs reside in specialized microenvironments or niches. So stem cell niches are sequestrated regions lined by supportive cells that regulate stem cell survival, proliferation, differentiation, and trafficking. By mid to late gestation, HSCs take up residence in the bone marrow, which remains the primary site of hematopoiesis throughout adult life. The bone marrow supports the maturation of all erythroid and other myeloid cells, and in humans and mice, the maturation of B lymphocytes. Unlike B lymphocytes, T lymphocytes do not complete their maturation in the bone marrow. Instead, T lymphocyte precursors leave the bone marrow and travel to unique microenvironments in the other primary lymphoid organs, the thymus. Then sites of hematopoiesis during fetal development. Blood cell precursors are initially found in the yolk sac, that means yellow, and then spread to the placenta. Then fetal liver and the aorta gonad mesonephros region before finding their adult home in the bone marrow. The mouse embryo is shown in 11th days of gestation, the human embryo 5 weeks of gestation. The sequence of changes in sites of hematopoiesis during mouse and human embryogenesis. Bone marrow. The stromal cells in the marrow that regulate HSC, quiescence, proliferation, trafficking, and differentiation include the following: endothelial cells that line the blood vessels, perivascular cells that are diverse in function and interact with endothelial cells. Then sympathetic nerves that transmit signals to other niche cells. Then macrophages, which influence the activity of other niche cells. Then osteoblasts, which generate bone and regulate the differentiation of lymphoid cells. Quiescent, long-lived HSCs are found in the perivascular niche, nurtured by perivascular and endothelial cells. Some HSCs remain quiescent, while others divide and differentiate into progenitors that develop into myeloid or lymphoid lineages. Then B lymphocytes complete most of their development in the bone marrow. Progenitors of B lymphocyte are found in the endosteal niche in association with osteoblasts. More mature B cells are found in the central sinuses of the bone marrow and exit the bone marrow to complete the final stage of their maturation in the spleen. Progenitors of B lymphocytes arise from the bone marrow HSCs but exit at a very immature stage and complete their development in the thymus, the primary lymphoid organ for T cell maturation. And bone marrow is not only a site for lymphoid and myeloid development, but is where fully mature myeloid and lymphoid cells can return. Many mature antibody secreting B cells, plasma cells, become long-term residents in the bone marrow. Some mature T cells also reside in the bone marrow. Whole bone marrow transplants, therefore, do not simply include stem cells, but also include mature functional cells that can both help and hurt the transplantation effort. With age, fat cells gradually replace 50% or more of the bone marrow compartment and the efficiency of hematopoiesis decreases. So formation, maturation and activation of T and B cells in primary and secondary lymphoid organs. Immature B cell, hematopoietic stem cell, T cell precursor, T cell maturation. So these three processes, immature B cell, hematopoietic stem cell, T cell precursor, etc. in the bone marrow. Then T cell precursor migrate to thymus for T cell maturation. Then later secondary lymphoid organs, T cell will be cell mediated immune response will be produced. In B cell, humoral immune response will be produced. And next is about thymus. T cell development is not complete until the cells undergo selection in the thymus. Miller, an Australian biologists worked against the power of popular assumption that the thymus was something other than a graveyard for cells. It was underappreciated organ, very large in prepubescent animals, that was thought by some to be detrimental to an organism and by others to be an evolutionary dead end. The cells that populated it, small, thin-rimmed, featureless cells, looked dull and inactive. However, Miller proved that the thymus was the important site for the maturation of T lymphocytes. T cell progenitors, which still retain the ability to give rise to multiple hematopoietic cell types, travel via the blood from the bone marrow to the thymus. In thymus, immature T cells known as thymocytes mature into functional T cells by passing through well-defined developmental stages in well-defined microenvironments. So thymocytes ultimately generate unique antigen receptors, T cell receptors, and are secreted to mature on the basis of their TCR reactivity to self-peptide MHC complexes. Then thymocytes whose TCRs bind self-MHC. Thymocytes whose TCRs bind self-MHC peptide complexes with too high affinity are induced to die, negative selection. And thymocytes that bind self-MHC peptides with intermediate affinity undergo positive selection and mature, migrating the thymic medulla before entering the circulation. Most thymocytes do not navigate the journey through the thymus successfully. In fact, more than 95% of thymocytes die in transit. Then thymocytes are tested for reactivity to tissue-specific antigens in the medulla. So thymocytes are also distinguished by their expression of two CD antigens, CD4 and CD8. The most immature thymocytes express neither to be as referred to as double negative. After entering the cortex, thymocytes upregulate both CD4 and CD8 antigens, becoming double positive. As they mature, they lose one of or other CD antigens, becoming single positive. Then CD4 T cells are helper cells and CD8 T cells are cytotoxic killer cells. Mature SP cells exit the thymus as they entered via the blood vessels of the corticomedullary junction. Maturation is finalized in the periphery where the new T cells, recent thymic immigrants, explore antigens presented in secondary lymphoid tissues including the spleen and lymph nodes. Secondary lymphoid organs. Lymphocytes and myeloid cells develop to maturity in the primary lymphoid system. T lymphocytes in the thymus and B cells, monocytes, dendritic cells and granulocytes in the bone marrow. However, they encounter antigen and initiate an immune response in the microenvironments of secondary lymphoid organs and tissues. Lymph nodes and the spleen are the most highly organized in the secondary lymphoid organs and are compartmentalized from the rest of the body via a fibrous capsule. Less organized secondary lymphoid tissues are associated with the linings of multiple organ systems, including the skin and the reproductive, respiratory, and gastrointestinal tracts, all of which protect us against external pathogens. These are collectively referred to as barrier tissues. And all secondary lymphoid structures contain anatomically distinct regions of T cell and B cell activity. They also generate lymphoid follicles, highly organized microenvironments responsible for the development and selection of B cells that produce high affinity antibodies. And lymph nodes. Lymph nodes is a highly specialized secondary lymphoid organ, most specialized secondary lymphoid organ. Unlike the spleen, which also regulates red blood cell flow and fate, lymph nodes are fully committed to regulating an immune response. Then they are encapsulated bean-shaped structures that include networks of stromal cells, support tissue packed with lymphocytes, macrophages, and dendritic cells. Connected to both blood vessels and lymphatic vessels, lymph nodes are the first organized lymphoid structure to encounter an antigen that enters the tissue spaces. The lymph node provides ideal microenvironments for encounters between antigen and lymphocytes and productive organized cellular and humoral immune response. Lymph node is divided into three roughly concentric regions: the cortex, the paracortex, and the medulla, each of which supports a distinct microenvironment. The outermost layer, the cortex, contains lymphocytes, mostly B cells, macrophages, and follicular dendritic cells arranged in follicles. Beneath the cortex is the paracortex, which is populated largely by T lymphocytes, but also contains dendritic cells that have migrated into the lymph node from the surrounding tissues. The media is the innermost layer and the site where lymphocytes exist, and lymph node through the outgoing deferent lymphatics. This is more sparsely populated with the lymphoid lineage cells, which include plasma cells that are actively secreting antibody molecule. Then spleen. This spleen, situated high in the left side of the abdominal cavity, is a large ovoid secondary lymphoid organ that plays a major role in mounting immune response to antigens in the bloodstream. Whereas lymph nodes are specialized for encounters between lymphocytes and antigens drained from local tissues. The spleen specializes in trapping and responding to blood-borne antigens. Thus, it is particularly important in the response to systemic infections. Unlike lymph nodes, the spleen is not supplied by lymphatic vessels. Instead, blood-borne antigens and lymphocytes are carried into spleen through the splenic artery and out via the splenic vein. Experiments with radioactively labeled lymphocytes show that more recirculating lymphocytes pass daily through the spleen than through all the lymph nodes combined. In spleen, which is about 5 inches long in human adults, is the largest secondary lymphoid organ. It is specialized for trapping blood-borne antigens. A stained tissue section of the human spleen showing the red pulp, white pulp and follicles. The splenic artery pierces the capsule and divides into progressively smaller arterioles ending in vascular sinusoids that drain back into the splenic vein. The erythrocyte-filled red pulp surrounds the sinusoids. The white pulp follows a sleeve. The periarticular lymphoid sheath pulls around the arterioles. This sheath is populated by T cells. Closely associated with the pulse are the B cell-rich lymphoid follicles that can develop into secondary follicle-containing germinal centers. The marginal zone, a site of specialized macrophages and B cells, surrounds the pulse and separates it from the red pulp. Then primary and secondary immune modulation. Antibody response to initiate antigenic stimulus is called primary response. It differs both quantitatively and qualitatively. Slow, sluggish and short-lived. Long lag phase and slow titer of antibody. Then predominantly IgM. Subsequent to primary response is called secondary response. Prompt, powerful and prolonged, short or negligible lag phase, much higher level of antibodies for longer period. Predominantly IgG. When an antigen is injected, an individual already carrying specific antibody, there is temporary fall in antibody level. Combination of antigen with the antibody. Duration of lag phase and perceived antigen vary with the nature of antigen. Diphtheria toxoid 2 to 3 weeks. Pneumococcal polysaccharide few hours. Non-living vaccine given in multiple dose. Then priming dose, first injection of antigen. Booster dose, subsequent injection of antigen. Live vaccine, sufficient for multiplication of organisms in the body which provide continuous antigenic stimulus.